Diverse tools for diverse coral reefs and cultures. Coral reefs house about 1/4 of marine biodiversity and provide sustenance for hundreds of millions of people worldwide, all the while protecting vulnerable shorelines from wave action, particularly under rising sea levels. Given their intrinsic value, it is a great tragedy that reefs are being decimated globally by climate change and locally by overfishing, pollution, and uncontrolled development. Despite this bad news, it needs to be remembered that stony corals are remarkably resilient and have persisted globally for over 200 million years. Therefore, efforts aimed at preserving these species need to understand the basis of coral health and resilience and develop tools and solutions that reflect this knowledge. With this goal in mind, scientists at Rutgers University and the Kewalo Marine Lab at the University of Hawaii in Honolulu are working together to apply cutting-edge molecular tools to coral science. The overarching goal is to bring this knowledge to the stewards of reefs at local sites to advance coral conservation.
Hi, my name is Lexi, or Alexi. I'm from the Republic of Palau in Micronesia. And being from a small island, I was lucky enough to be able to go out on boats with my grandpa when I was younger. And that was the first time I really saw what a coral reef was and how beautiful it was. And, all of the fish and other animals that it housed. And it wasn't until I got to college and came over to Hawaii that I really realized how much I wanted to study corals. I think it was after seeing my first coral spawning and watching fertilization happen under a microscope, and just thinking that these tiny little beings can someday grow and settle and create whole ecosystems that, islands of people depend on. It just made me want to study them, figure out how to protect them, all with the goal of, preserving these kinds of environments for future generations.
Kewalo Marine Laboratory is a research unit of the University of Hawaii at Manoa. And we do marine research on a variety of organisms, particularly those that are indigenous to Hawaii and to the region. And we do everything from molecules to ecosystems. We're very keen on collaboration. And so being able to work with groups like those at Rutgers, we have a home field advantage of living on coral reefs and having a great seawater system. But we really thrive on these kind of collaborative opportunities to work with people with special skills in molecular biology, especially the omics, genomics, proteomics, transcriptomics, metabolomics, of bringing really cutting edge techniques to the classic ecology and physiology that we do very well here as well.
Some of the specialized tools that we use in our lab involve extracting the chemicals called metabolites from corals, identifying the expression of genes. This is called differential gene expression analysis. Cataloging the proteins that are expressed under stress, as well as looking at the genomes of corals to identify the stress response pathways and to combine these very different types of data sets into a coherent picture of coral biology and evolution.
So our work has found in a single bay in Hawaii that there are two groups of Poeciloporacutta. There are those that are diploid, that have 2 copies of each of their chromosomes, and those that are triploid, that have 3 copies.
So this extra copy of the gene can cause changes in cellular physiology, like changes in immune response, changes in cell size, and other species-specific changes.
We have also found that the triploid individuals have spread throughout the bay via asexual reproduction. This means that they have spread clonally, so they are all genetically highly similar.
So we're trying to identify which processes that are differentially regulated in these triploids can allow corals to be more resilient to climate change.
These cutting-edge omics techniques will be used to develop tools that can be easily used in the field by reef managers to assess coral health on their local reefs.
So 2 of the tools that we used are strip tests and antibody tests. Now the first test that we're developing is test strips, which are designed to assess human health, for example, through urinalysis. And we've adapted them to diagnose coral health. Now these AccuTest strips have 10 tests, of which 2 work for corals, ketones and leukocytes. The ketones measure ketone bodies that are a signal of energy deprivation in corals, And a strong test strip reaction means that coral is energy deprived and under stress. And the other strip test reaction that we use to measure coral health are the leukocytes, and these leukocytes measure the immune reaction in the corals, and the stress also shows a high reactivity using these strips. These strips can now be done using a smartphone app that we've developed. It's Test Strip Diets and is ready to be used in the field with only a few inexpensive reagents and a special smartphone holder, which makes it really easy to use in remote locations.
So my research looks at the effects of microplastics on corals. Microplastics are a hot issue in the news right now with people finding microplastics in human blood. And so when you get a piece of microplastic and you put it in a water, so for instance in the ocean, it does one of two things. So the first thing, if you think about all the microplastics that you use in your daily life, so think about microplastics we use for kitchen utensils, think about the plastics that are used in the building of your car. We use microplastics for a lot of different things. In order to do that, a chemist has to give it a certain chemical to get a certain desired property. And so when you get a piece of microplastic in a body of water, it acts like a sponge. So one, all those chemicals that are made of, it'll dissolve into water, but also it picks up any of the chemicals that are free floating in the ocean already. So I'm particularly interested when you get coral, which have recently been found to eat microplastics, what happens? Do the chemicals leach into the coral? Maybe the chemicals aren't an issue. Maybe the corals get rid of the piece of plastic before the chemicals have any effects. I want to understand how microplastics affect their health, their survival, but also reproduction and some of the implications for future generations.
I'm really interested in this place called Mermaid Bay, and it's really special because it has reduced pH and it has a little bit warmer water than outside. So, when you think of a place like that, you shouldn't really be seeing coral reefs live there because it's a pretty stressful place to be. But what's so amazing about it is that there's lots of diverse, different types of species. So part of my project is looking at one small species of coral and going to different sites in the bay and taking proteins from them and trying to see what kinds of proteins those are. One thing I'm learning here at Kewalo is that protein expression and diagnostic tools like these are ways that we can identify how these corals are being stressed out. And by doing that, I'd also like to try and figure out, why these corals are able to live in these stressful conditions that no other corals can.
While it's important to understand coral health on an individual level, it's also important to understand how a reef will behave on a large scale.
My work with the Kewalo Rutgers group involves incorporating data regarding population genetic structure of Lantipora around Hawaii. And I do that through mathematics and computer simulations. We hope to put this together in a theoretical model in order to understand the impacts of climate change on Lantipora. And we'll be able to identify sites that are particularly vulnerable and resilient to climate change.
Coral research and conservation have expanded greatly on the world stage, with approaches including ecology, climate science, and modern omics methods, as described here. However, much of the work is being done in labs in the US, Europe, and Asia, where no local reefs exist. This has led to a disconnect between mainstream coral science and the caretakers of reefs, who often live in small island nations and do not have access to the resources needed to take part in this scientific revolution or to reap its benefits. For this reason, the Rutgers Kewalo Group considers on-site training of local experts in the generation and interpretation of scientific data as their most important goal.
And so for that reason, when we do our work, it's always focused in not only on the organisms, but on the people who depend on the resource. And as a result of it, we do a lot of work with managers, what we call so what science. We do a lot of science, which is our primary job, but we always ask the question, so what? With that knowledge, how can it be used in ways to address some of the pressing problems, not only today, but especially as a legacy for future generations?
So one thing I think that's really important about the skills that I'm learning out here, along with all of these collaborative projects with researchers from all over around the world, is the ability to bring these skills back to my home island and other islands in Micronesia and build capacity there. Because the kind of work that I'd like to do is one that benefits my own community and communities that depend on these coral reefs. So I think one really important thing about all of these skills is, being able to apply them there and do the kinds of research that the people will benefit from. That like they can take this information and be like, okay, I can make these kinds of, like, I know this about the coral reef right here, I'm gonna make these kinds of laws, or local chiefs can make decisions about their coastal areas that can help with conservation and, preserving the resources better.